So, ladies and gentlemen, today I'd like to talk about something related to biology, but from the data mining direction. Today, we know that the advances of bioinformatics with the significant implications to biotechnology and healthcare have a profound impact on the personal and collective life for decades to come. With the advent of the next generation sequencing technology, we can acquire data fast and big. But the intermittent wet lab and also the bioinformatics lab cannot keep pace with the development of the data. So therefore, we need efficient method to get knowledge directly and fast. VMine is the technology we have developed for many years. It is a proven technology for pattern discovery, synthesis, and analysis of biomolecule sequences. Now, let me give the rationale behind. When I say young faculty, I begin to ask the questions. If there's some information embedded in some data, can we find it, okay? Without prior knowledge, without any clue, can you find it? Then what does it mean, okay? What does it mean from the entropy sense? If they are random, they are independent, they are equal probable, definitely nothing you can find. But if they contain meaning, if they are organized, okay? If I can find out how they deviate from the default random independent model, then at least I know that it contains something. You cannot consider it, right, to be arisen from a random situation. And from that, we begin to search a way. First, what is information? Can we know information here and there? And then we go one step further to find out not only a measure, but what exactly. Now, for example, suppose you are given a sequence of very long sequence, 500 million characters. If it contains some short sequence that have meaning, can you find it, okay? This is very simple. Now, actually, I can give you a demo, but actually, because of time, within less than 2 seconds, we can find out some non-random association of local sequence. And what are they? In this case, like pride, fortune, and knowledge in one of. If I ask you, do they contain meaning? Unless you do not know English, you know that it contains meaning. Why it is there? Why we can find? Because there's an English, there is a phonetic, there is a language structure behind language. So what are they? Actually, when I first discovered binding site, my student asked me, how do you know that it is important? How about, let us begin with some English example. So we take the whole text of Pride and Prejudice. We remove all the punctuation, all the spacing, no uppercase and lower case. So it's actually is a 500 million long character sequence. Again, within 2 seconds, we found out that all the non-random, not independent kind of unit. And it contains the data, the language structure, phonetic, and everything, all embedded. There are short sentences correspond to English words and phrases with language structure and implicit meaning. So when we apply the same technology to DNA, okay, then we can find out the functional unit would contain some kind of physical meaning, chemical meanings, biological meaning, structural meaning, functional meaning. Why? Because they are encoded for that purpose. So when we apply this to the upstream of the transcript factor of DNA, then out of the 18 data set, we found 13 of them. They are actually the binding site, very high up. We also apply this to transfer RNA. We all know the transfer RNA is a well-structured. Again, without any prior knowledge or clue, we are able to find out all the pair stem. And beyond, we can also find out what we call a distant pattern. It means that they co-occur together in a molecule. Why they co-occur? Because they are responsible for tertiary structure, folding, and many other things, okay? It's embedded there, it's encoded there. So of course, we do not apply to trans-RNA, right? We have learned a lot about trans-RNA, but we can also apply to other ribosomal RNA. The whole idea is no matter which is protein or RNA, DNA, or language, as long as there is a functional meaning there, we will find it okay. So therefore, we propose a new paradigm. How can we move data into knowledge? By data, definitely, in a bioinformatics setting, it could be biosequences, can be a microarray, can be a table. By pattern, we mean some association, okay? They are not inter-association or relation or rule. And if we represent it in certain form, so that it can be verified from the real world, okay? Through experimentation, through scientific application, explanation, so that we can infer the structure and function, and perhaps if we do not know those before and we find it and explain it, then it becomes scientific discovery. Now, there I want to give you a briefing of the technology behind. Now, first, protein sequence. We all know from a primary structure to secondary to tertiary and then to quaternary. But the current state of art is easy to sequence, particularly with the new next generation sequencing machine. But hard to crystallize, and reverse mapping is very difficult from a 3D and going back to the primary structure and tell you here, right? It's not so simple. So we might try to discover the critical patterns and region and relate them back to the structure and function to shorten the search process. We help biologists, not replace biology. Now, we can using unsuper. Unsuper means that you do not need prior knowledge. Super means that if you have prior knowledge, you can use the prior knowledge. We begin with some kind of multiple sequences. And then we discover and prune the pattern. By pattern, it means that they are sequence of association, not only pairwise, the whole word, okay? Then we align them and cluster them. We bring the similar pattern together to reflect the local functionality. And then, of course, we can go more further into the structure. And then we can further partition the so-called aligned pattern cluster into subcluster. When we bring something functional together, there are two issues. One issue, what are the functionality that govern that region? And other, where are the discriminative information you can get to reflect the protein classes, protein group within the region. We localize it so that we are not talking about a whole molecule. We zero in to find out the functional region, like a word, okay, or a similar word. And then we can use information to show are they related back to the class characteristic of the protein. If we know that at least we can explain, we can find some. And then we want to find out what pattern in those regions they co-occur on a sequence, and what pattern in certain regions are interact with another region. There's a chemical kind of bond. If there is, then we can find out the interacting site. We can find out the site with the chemical, and then we become a drug discovery. So the whole idea is once we find out, zero in this short sequence, then we can relate this back to the real world, the structure and function and protein classes. And then we can enhance it, but from we bypass the bottleneck. We don't need any prior knowledge. Sometimes it's the prior knowledge, the prior requirement that costs us time, try and error, okay? So now we have 5-step synthesis. First, we discover and prune pattern. Second, we align and cluster pattern into align pattern cluster. And then we partition them into distinct subcluster to reflect their discriminability. And then we use information measure to review the classes so that we have objective basis to show what are important in what way. And then we discover co-occurrence and also the interacting APC plus chemicals. Then, now, give an example. Suppose we have a long sequence, just character, we don't know what they are, right? But by pattern, we discover some association, wherever they are. We don't need to a consensus region, right? As long as they are similar, we can discover them. Then we remove the redundant pattern. By redundant, if the super pattern contains a sub-pattern, it's redundant, and vice versa. Now then we align them. By alignment, it means that we bring them into some kind of common functional framework so that we can see the commonality among these patterns. And then the APC contain correlated non-random pattern implying similar underlying structure and function. And later we found out that, yeah, it did relate it back to the structure and function of the molecules. Then we further partition them into, and then using some information measure to show that they are indeed can reflect the subtle class characteristic. For example, now we have an aligned pattern cluster. And if we bring back the ground truth, then we can find out which pattern, right, from the local functional group. Now we look at local functional. Then within this local functional group, which pattern are corresponding to what class? Then we found out that some patterns are connected to mammal, some patterns are to high plant, okay? So this is any sequence, right? Because the association of related back to the mammal. If you look at the column, in an unsupervised way, we don't know what is a class, but if we find out certain site which is high interdependency with all the other sites, right? There's information measure can pick up those sites, and you know that those sites contain amino acid which are related to specific class. Even class label are not there. Because what is a class? Class means that you reflect something that interdependent, right, into certain group, okay, certain functional group. In a like manner, even amino acid, we can go to amino acid level. If you ask me, for this important site, which amino acid will be related to what class? Or furthermore, we can find out which subgroup will belong to which class. This subgroup belongs to mammal, and the other group belongs to plant. So that by co-occurrence, it means that if two patterns, they always co-occur on a molecule, it indicates that they have some kind of joint functionality. It may govern their folding, their interaction, or any way. So here we find out that these are co-occurrence APC, but these two are somehow co-occur. If you go at the subcluster level, we also find out that these are co-occur. You can see that two co-occurring groups. It suggests that two groups of molecule, right? Their function, the co-occurrent functionality could be a little different at a distance. So the whole idea is we suggest some joint functionality. When you're using the Raptor or other program, then you can see how those regions are related back to the structure and function, interacting energy, whatever, right, of that. Now, in a like manner, we also found out that some co-occurring group related to mammal, other related to plant, okay? So in a nutshell, this is the methodology we have developed and we unify. We can using co-occurrent measure to bring out those APC which is co-occur. We can also using another kind of score, like a chemical interaction score, to bring out those regions that can have interaction between protein and protein, protein RNA, or between protein and chemical, and so forth. So now we give some case study, then I will end my talk. Sorry. This is something. Now, application. Now, in the classical, in the traditional method, so we have begin with a lot of data. Now, in our method, okay, we discover prune and align and cluster pattern to bring the similarity functional unit together. And then we review the subcluster, review the class, co-occurrence, and also biochemical interaction. And then we pinpoint the critical target domain for wet lab experiments so that we can pass it on to the biologists to zero in, right, to confirm. So this scientific method, right? Now then, so now we begin with three. First, we will demonstrate, using cytochrome-C, demonstrate that this can be applied to genetic information research. Second, the drug discovery. And thirdly, in the disease treatment. So I don't have to say more about cytochrome-C. It is very important. It's binding and release from the mitochondria, prevents cell death for cancer treatment. So this is the way we can find out how things could occur. Now here we demonstrate how can suppose so don't know what happened. Now, yeah. So you lose the animation. Oh, sorry. Excuse me. Yeah. Sometimes you rely too much on high tech. High tech can betray you. Okay. Now here, first we found out that these are occur, and then we found out that they're embedded in there in your APC are some significant site, which is very significant within this site. And both PFAM and POSI cannot find this because they only find M, but we find M embedded in something, okay? Now then, for example, in a superhighway, if I bring back the class label, then I know that using some information measure, and this pattern, right, just pertaining to a single group, which is high plan mostly. And this group pertaining to mammal, because the entropy is 0. And some, they are pertaining to more than one group, okay? It means that these patterns are shared by 3 different groups, and one are specific for one group and the other group. Automatically, then we can let the biologist figure out why, okay, and then perform experiment to verify. Now, suppose without any prior knowledge, with some kind of information measure, we are able to find out certain site, right? They must contain some very distinct information, like this one, with very high interdependency measures. So for this group, all E and A, they share with mammal. Some with the insect, some with high plant, and some with that. That automatically we are able to pick it up into some kind of class, okay, and reveal the underlying functionality of each class. Even we can go into the amino acid level. So I can skip this. Now, in a nutshell, we are able to, say, find some kind of co-occur APC. We can find it, map it into the graph space, in the pattern space, and also in the data space, and back into the 3D space, okay? So this is more very, very interesting. For example, once we find out this cluster, then we can find out that actually this group contains interlaced pattern. They span different domain, okay? So their major functionality are a little different because, and then the next one we go into the drug discovery. Now this molecule related back to the curing of cancer. I do not want to go. I do not like to go to the detail, but that's the same. By the same notion, we are able to discover some of the pattern, and then we can associate back, right, to this. You can see A one class, E another class, and all that. And once we are able to isolate this functional group, then there are some unknown features, okay? We can associate unknown features to which group? Like the 16 features are associated with to set 2, okay? And we call it semi-supervised. We're using unsupervised to find a group, and then using that to pick up something related to that. Suppose if this is a cancerous data, right? Then once we don't know what does it mean, right? If you can find out certain group, then you can bring in other. So this is just further demonstrate. You can find out the interlaced kind of co-occur pattern and relate back to the 3D well. Now finally, we just show that we can also apply this to find out the region, the functional region in a molecule from the APC we discover. Now these are two very interesting molecules. One is longer, the other is shorter, but the whole functional group is here. So again, there are two classes, but these are scattered along the long sequence. They're 500 bases long. But once we find out these 3 APC, you can see the differences very easily. Okay? Now, if you want to plot the APC on the long sequence, you can see they span different domains, okay, at a distance. Why? Because structurally, functionally, there are some intrinsic meaning there. So that we certainly, I'm sure the biology will be very happy to find out why. Now, just further demonstrate. From now on, I just show you we are able to discover something validated by biologists and from biological papers and so forth. Okay, right. You can see the key thing are picked up and then relate back to the 3D well. And so are these other group. And here is very interesting. Lysine, they are today we do lysine are very much related to cancer, right? And only our method can discover all the lysines spanning in this. You can see that we pick them up, and two K are found in the significant functional association. And then of course there are many more. Now, so I want to conclude. That's what 2 minutes. I will conclude. Right? Let's see. Now, to conclude, and now it's lawful and unique, right? We are algorithmic based, no ad hoc, and robust, direct, and efficient, and based on statistics. So when we talk about pattern, those are 1 out of billion, right? Chance it cannot be random. Better than existing method, fast. We have to do a lot of serious comparison in time complexity, in space complexity. And then they are data-driven, not rely on prior knowledge, avoiding trial and errors, experimentation, resulting in great reduction of time and cost. And fast in processing in large volume of data, linear time, performance in discovering non-redundant patterns. So by bioinformatics, we can reduce time from trial and error exploration, reduce material and labor costs for wet lab experiment. In drug discovery, a deep understanding of target at a faster pace, okay, because we zero in from a lot of the ocean, you get you the island, then you can look at the island much more easily, easier and quicker in target identification and drug discovery, and study diagnosis and treatment, that we can have the diagnosis protein involved in the immune response, improve research efficiency by ordering a faster processing time and lower false positive rate. So, in summary, it is a unique process to allow biologists and medical researchers to screen out protein and RNA and DNA target region, pattern, site, and residue to pinpoint the critical functional domain common or different to sequences associated with inherent classes for research and pharmacological and healthcare applications. And ubiquitous all-in-one user, we can all back into one GUI, right, for the user to use for visualization, interpretation, that reduce time, labor, and cost in discovery and analysis process. And we have definitely, we can use it for documentation. So this is one of our public paper, and this is acknowledged to my group, okay. Thank you.